Okay, so we actually talked about this smart technology. We saw a video how this smart actually works, uh, and
Yeah.
this is actually a very similar to Illumina sequencing, right? Very similar to Illumina sequencing,
Yeah.
but the main difference lies in the fact that this smart technology actually moves into the atomic level, and it actually sequences your the whole DNA by nucleotide to nucleotide based, and that's how it's highly error free mostly, and it can be conducted in parallel with multiple chips. So in I think at a single time you can sequence almost up to thousands to 2000 number of DNAs on a single flow cell.
Mhm.
Okay, so that's the beauty of this
Yes.
technology. Now we talked about actually what it has is it actually has the some ZMWs, right? So these are actually zero
Yes.
mode
Yes.
waveguides. Okay, so this
Mhm.
ZMWs they have some nanometer scale diameter, and these actually
Okay.
provide the spaces for the parallel sequencing to take place. And to record the
Okay.
DNA synthesis in real time, the focus of the camera needs to be kept constant, and therefore we have to avoid the requirement those that are required for minor adjustment through the whole sequencing procedure. And therefore, to maintain these two conditions, the physical position of each incorporation event should be predetermined and fixed during the synthesis. And to
Mhm.
achieve this, DNA polymerase instead of the template is actually immobilized inside the ZMWs. Now, the template that you have, since the polymerase is actually fixed, so the template that you have, it can be either a circular template or a linear template, right? Now, the linear template generally generates a single pass long reads. The long reads might vary up to 10 kb, while this
Okay.
circular template, they generate multiple pass but have shorter reads of almost 250 base pairs. Now these templates are generally held by DNA polymerase. So if you have some bacterial DNA, then obviously you will have some lower reads because they are circular DNAs, right? Bacterial DNA is generally a circular DNAs. Whereas if you
Yeah.
have some other DNA from a multicellular organism from other sources, then you know you have the linear DNAs that you can use. Now, with this particular a fluorophore with a unique color is actually linked to all of the four DNTPs through the triphosphate chain, and
Okay.
this omits the extra cleavage that is required for the DNTPs with dyes attached to the bases. And by constantly providing the fluorophore level DNTPs at high concentration in the reaction solution, this performance of this polymerase, including the enzyme's accuracy and processivity, is actually greatly enhanced and improved. Now, the size of these ZMWs that you have, they are actually very small. Okay, and this is really too small to hold the free fluorophore level DNTPs, and the
Okay.
duration for each DNTPs that stay within the detection distance is generally determined at tens of milliseconds, and which is really short enough to cause only low level of background fluorescence, and
Yeah.
therefore the noise is generally eliminated.
Okay.
Okay, and apart from that, any unmatched DNTPs that they have is obviously quickly diffuse out of the ZMWs because it is really small. It cannot hold the DNTPs for a much longer time. But
Mhm.
the complementary DNTPs, they are generally held by the polymerase during the incorporation event, and this resulting pause that whenever the DNTP is actually added is being incorporated, the polymerase and the DNA stops moving. They're just for a fraction of second, but it actually stops moving, right?
Yeah.
And
Mhm.
this pause actually leads to the extended fluorescent signal, and the type of the incorporated DNTPs can be identified by the unique fluorescent emission spectrum. So when complementary DNTP is being added, it actually fluoresces for a longer period of time because of that short pause. Whereas if it is not incorporated, then it simply diffuses out. So therefore, it cannot give a very bright fluorescence. Okay, and that's how this distinction is actually made. And when the DNA is incorporated, a phosphodiester bond is created, and once this phosphodiester bond is created, the diphosphate that is attached with the fluorophore is actually cleaved off by the DNA polymerase at the end of the incorporation event. And this released fluorophore, they quickly diffuse away from the ZMWs, and it restores the fluorescence signals back to the background level. Okay, and as you have seen the whole thing, that actually how it occurs, and using this particular strategy, this strand synthesis is actually performed continuously and signals are captured in real time. Now there are multiple sequencing steps, including focusing, signal scanning, washing, and releasing of the fresh DNTPs. And they are actually removed in this novel technology, and they all greatly shorten the sequencing time. And by mimicking the natural synthesis procedures of DNA polymerase to maximally extend the DNA, the speed of this strand elongation is much higher than those of any other technologies. And this enables the generation of very long reads that are actually kilobases in length. Okay. Now, there is actually another very great attractive feature of this smart technology, and that is the ability to directly sequence and detect the methylated DNA without the need for conventional bisulfite conversion. And generally, there are three types of epigenetic modifications that regulate various biological processes. Majorly three. These are DNA methylation, histone modification, and chromatin remodeling. Right? These are the basics, main type of epigenetic modifications. And these actually mostly control all of the various biological processes like gene expression, genomic imprinting, including the X chromosome inactivation. So any defect in these processes, it has been found that these defects in this particular process have been linked to various human disease. And mapping of these genome-wise methylation profiles, it actually has been carried out in different research areas by obviously using various methods to enrich the methylated regions, and then it is followed by massive parallel sequencing such as methylated DNA immunoprecipitation sequencing. In short, it is called MEDIP-seq. Okay. Apart from that, you can also go for MBD-seq, that is the methyl CpG binding domain sequencing, and of course the easiest of them is actually bisulfite sequencing, right? And all of them actually require the additional treatments during the library construction. But in smart technology, the detection of an incorporation event is actually characterized by two parameters. What are those? First is the pulse width, and second is the interpulse duration. So what is pulse width?
Okay, so sorry, what was that again? I didn't hear you.
Yeah, actually there are two parameters that actually that characterizes this incorporation event, or that controls this incorporation event. First is the pulse width, and another is the interpulse duration. So what is pulse width? So pulse width actually measures the duration between the binding of a nucleotide with the template and the cleavage of the fluorophore associated phosphate group. Okay, it actually calculates how long. So the first event is actually that the nucleotide is bonded to the template, right? That's the time. So the time starts at that point till the point when the DNA polymerase cuts off the fluorophore. This is the pulse width. Okay, and what is interpulse duration? Interpulse duration is simply represents the interval between two successive pulses. Okay, the time between two successive pulses, that is the interpulse duration. Now, a proof of concept study that actually proposed that this methylated DNA can be readily detected by the smart technology, because the kinetics of DNA polymerase was actually predicted to be sensitive to DNA modifications, and therefore they will generate very distinct profiles for both pulse width and interpulse duration when it is compared to an unmodified template.
Okay.
So, and actually the results demonstrates that various different kind of methylation pattern, for example methyladenine or methyl cytosine, or in fact hydroxymethyl cytosine, and they were actually readily detected and distinguished from the unmethylated controls based on their unique pulse measurement. And it is seen that when you need to add a group against a methylated DNA, then the interpulse duration generally exceeds. Okay, and how much it exceeds? The time that is taken to incorporate a base against a hydroxymethyl cytosine or against a methyladenine or methyl cytosine are all different. They are all different, and by studying this difference of times, you can actually state that which bases are undergoing which kind of methylation. Okay, and along with all the other advancement that is offered by the smart technology, there are actually some challenges as well that needs to be addressed. The first is actually the error, because if you have almost suppose you have 150 bases. Okay, suppose you have 150 bases, and then only it is seen that only among 150 bases only 130 base can be aligned correctly to the reference, and this corresponds to a drop in sensitivity, right? And among this, sometimes indels, the insertion and deletion, they actually account for almost 74% of the errors generated. Now, there are actually several solutions, such as increasing the pulse width, increasing the signal intensity, and they actually have been proposed to improve the accuracy. And another effective solution is to actually sequence the circularized template molecule multiple times. So if you sequence a circularized template molecule multiple times, what will happen? It is going to dramatically increase the sensitivity, because most of the errors that will generate that are generated stochastically, then the probability of having those two errors occurring at the same locus is greatly reduced with increased coverage, right? And although there are more improvements are pending to fine-tune this detection system and increase the accuracy, the smart technology actually has a very advanced MPS, that is multiple parallel sequencing, and it is actually provided by the single molecule based high throughput information at both genomic and epigenomic level. And this actually makes this smart technology a very attractive tool for sequencing. And this mapping of this genome wide methylation profiles has been carried out in different research areas, and people have actually done and found these epigenetic modifications can be very well understood by the smart technology rather than by using solid or by the ROS system and everything else. So if you are into finding about this mutation, then of course you need to go for this, or to epigenetic modifications, then you of course you need to have this your sequence DNA sequenced by this smart technology. Okay, and this is actually the whole smart technology that is present right now, and this is actually provided by this Pacific Bioscience technology. Okay, now generally now there are actually variety of MPS platform that exist that coexist in the market. And this is actually,
Okay.
this whole chart is actually a cross comparison between them. And this might facilitate the platform selection that is based on application or the reported performance. Now, while the second generation MPS technology, they rely on the detection of these cluster generated signals. This third gen MPS technology, they capture the signal that is emitted by the single molecule, and that enables,
Okay.
and this enables the direct sequencing of this DNA and RNA molecules at high speed.
Mhm.
And furthermore, by increasing this read length from tens of bases to several kilobases, this
Mhm.
3D MPS based de novo assembly and this indel detection will be dramatically enhanced.
Mhm.
But the prevailing use of this second generation technology in this biomedical and diagnostic field, they will continue due to their stable system and much lower error rate. Because
Yeah.
as I am increasing the throughput, my error rate is also going to increase, right?
Increase.
Because there's a kahavat in Hindi that is shaitan ka kaam jaldi ka. So that means, yeah, so whenever you are doing something in a bit hurry, you are going to make some mistakes, right?
Yeah.
So that also happens with the 3D technology. However, they are very smart, they are very fast, but what they lack is that stability and this high low error rate. That are the things that are lacking in this third gen thing. So this table actually shows a comparison of this whole platform performance using commonly adopted metrics. And with the arrival of this MPS era, single gene studies actually have gradually evolved into a panel and genome wide studies. And this
Okay.
transition challenges the bioinformatician to improve the algorithm for resequencing based read alignment and also for de novo assembly of unknown sequences.
Mhm.
And computer scientists are also need to be brought into the loop, and they need to maintain and update the public database, right?
Mhm.
And finally, it's up to the biologist to interpret this filtered result and follow with the functional studies. And
Okay.
as feedback, this unsolved biological puzzle will guide the engineers to continue to enhance the performance of current MPS platform or invent
Mhm.
fundamentally novel technology that are actually aimed for
Mhm.
new applications, right?
Yeah.
So this MPS rail will simply, obviously, they will continue, and as our advancement for this understanding of this human genome and unsolved genetic disease goes on.
Mhm.
So now the question is actually that why NGS analysis is different when you to analyze the data, because the idea of sequence analysis is actually familiar to most of us,
Mhm.
but the challenges of this NGS are forcing some new thinking and new strategies. And this is an actually active area of research, and the intention of this particular slide is to review the issues and strategies that are applied to DNA sequence analysis by NGS. Now, the key difference of this NGS data can be summed up in just a single word, and that is extreme. Why? Because
Yeah.
a single NGS run, it simply dumps about 100 million nucleotide onto your hard disk.
Mhm.
So, and you will need almost 30 runs or almost 120 million reads to cover the entire human genome at
Yeah.
a 1x coverage. At a 1x coverage. Now, the Human Genome Project, it required 15x coverage. But the current opinion is that 30x coverage will be required with the shorter NGS read. So that means you will need almost 900 runs or 90 billion nucleotide or almost 200, 150 GB to 200 GB of FASTA format data on your hard disk to resequence a single person. Now, a personalized medicine database with the assembled genomes uh, for just only 10% of United States population, it would actually hold almost 100 TB of FASTA format data to be searched. And when you have data set of this size, we encounter several significant problems. And these problems might vary from input output problems. It might be the problem with file and directory size, because there are certain limitations in current operating system, right? And simple searches can actually take years, and many tasks are actually just not possible with the traditional tool. So this NGS produces short reads that are frequently searched against the large chromosomes, and each raises a particular problem. Now every read or every entity, they all require a particular header to be established in the memory. And because NGS reads are so small and so numerous, massive resources can be tied up simply establishing these headers. Because for each DNA strand, for each DNA fragment, I need to write the name of the fragment size, which the gene to which that following fragment belongs to, the location of the loci of that particular fragment, right? And the location of that, or the map of that particular fragment. So these are the things that are actually must be present in the header, because you need this data once you need to align this data to a reference genome, right? So these are actually so finding these and generating these numbers are actually a very enormous task. And so what they actually do, so if you have a single large sequence that cannot be segmented without the complex data handling, and then all the interactions over the entire length of that sequence must be held in memory as a group, right? And optimizing for one of these extremes often makes some significant compromise for the other. And the NGS reads often include the errors. Now the methods that are actually sensitive enough to match all or most reads are usually computationally intensive, and they also find too many false positives. Now there are actually some methods that are discrete and efficient enough to complete in a relevant time and find few false positives, but unfortunately they also find too many false negatives. And with almost 3.6 billion reads, a small rate of false positive can actually overwhelm many real matches. Right? So for this alignment, often you have to use BLAST, right? And we are going to study alignment. We are going to see some servers that can perform this alignment, how to align, how to develop the evolutionary tree and all. We are going to see that in a later period of time while we do the proteomics part. Okay. Now this BLAST, they often lose the near perfect matches of these short reads due to the way these scores are calculated. Now generally the score that is used is actually the S-Saha score. This S-Saha based methods, they actually build arrays with a sampling offset that is equal to the word size. But with the short read, this result is really insufficient sampling in an array to detect many perfect matches. Apart from that, there are some slim search areas that are actually very essential for NGS data. Now, many part of this analysis, they actually can be done more efficiently if the read lengths were consistent. Now, there are some researchers who are trying to adjust the samples to constant read lengths by culling the outliers or filling in shorter reads or assuming the longest sequence as the universal length or grouping these reads into a discrete size groups. And typical NGS analysis is actually concerned more with the correct matches, and that means and less with the distant relationships, right? Now, because this NGS, they often surveys many closely related species or many individual of a single species, the discriminating close relationship, they actually become more important than the distant relationship. And there are some repeat regions, and these repeat regions are conceptually no different. But the shorter reads mean that the number of repeat that require the secondary data to resolve increases dramatically, right? Because the repeat lengths are huge, and even in the NGS we have shorter reads. So that means we have to cover much more to finish up a particular repeated sequence. So this and now generally a typical NGS analysis pipeline might involve a filtering, base calling and sequence alignment, followed by the assembly or other downstream task. This filtering and base calling is usually done by the manufacturer's software. Okay. And those are sometimes enough, those are sometimes good enough to give you a very excellent result. But there are some people who are working on de novo assemblers for NGS, and generally the focus is on remapping the reads. Now the basic idea of sequencing search is to find the matches by strength of similarity. But this computer, on the computer where we work, they can only work in 0 or 1. That means either it's a match or it is not a match. So therefore in computer this similarity is not allowed. So that means if you have a protein, suppose you have a protein. Now in that particular, suppose you have a gene. There are two genes. Gene A belongs to a species A, and gene B belongs to species. Gene A belongs to species B. Both the genes are similar, but there are minor modifications. Now when you sequence both the genes, you find that yes, there are certain modifications, there are certain mutations in between them. But when you see the translated protein, you see that a similar amino acid is being replaced by a similar amino acid. That means a positively charged amino acid, generally lysine, is being replaced by arginine. Now in respect of this, in respect to this chemical environment that a particular amino acid carries around it, its surrounding is similar, because arginine and lysine both have similar kind of chemical environment, and both of them can perform the similar kind of reactions, that is binding to a negative zone. Since they are electropositive, they generally go and bind to the electronegative zone of another protein or the DNAs and all. Okay. So this is a similar mutation. But if the computer does not understand this similar modification, it only identifies that it is a modification. So it is going to say that it is a change. But in real life, it is not a change. Because those kind of mutation generally occurs in our body. And since these are similar mutations, generally people and we overlook this because it does not make any contribution. It does not change the phenotype of an organism, right? So the link between, there is no link between that kind of mutation with genotype and phenotype. But with NGS, it is going to say you that there is a link between the phenotype and genotype, because it does not understand the similarity. It only understands there is a match or there is no match. So the typical strategy that people have developed is to chop the sequence into sections and look for the fragments of identity. And Smith and Waterman was actually a scientist who developed a program called DPM, that is the dynamic programming matrix. And it actually breaks the sequence into smallest possible unit. And what is the smallest possible unit?
It's the nucleotide.
Nucleotide, right. So it actually breaks the sequence into individual nucleotide, and it plots the highest scoring diagonal through the matrix of short exact matches. And this method actually remains the gold standard. But unfortunately, it displays some, it takes into some account of space-time complexity, and it is not possible for task of any size. Okay. And apart from that, the great innovation of this blast was to look for the larger chunks. Okay. And search in an one-dimensional array. But as a result, the blast displays the space and time complexity. But though not as precise as is DPM, it is close enough, and and the more important factor is that the blast is fast enough to be practical. Now this blast uses exact matches of generally 11 nucleotide to anchor a local DPM, and all the final data are generated by the DPM. Now, while the blast method has many advantages, it also has some limitation, and some of these limitations are very critical for NGS. Now, there are several authors who have proposed newer strategies for building, scanning, and processing the data from the arrays of short exact matches, as well as for the strategies for eliminating the need for this DPMs. But computer science teaches that the law of physics constrain how you can build an array. Programming languages, however, they contain a variety of methods for working with data in that array structures. So it is actually possible to create novel permutations by combining aspects of this method as well as with very clever data handling. Okay. Now in order to properly interpret a sequencing result, the spectrum of sequence variants needs to be carefully classified, and generally there are actually there's a standard for interpretation and reporting of sequence variations, and the practical by taking into the practical experience in various people's laboratory and all, the sequence variants can be classified into 6 categories. The first is the reported pathogenic mutation. Okay. Now these kind of variants are actually, these are the sequence variation that has been reported in multiple unrelated patients or unrelated families, but they have to have some clinical correlation, okay, or some supporting functional studies, and actually this mutation is a recognized cause of the disorder. So this is a reported pathogenic mutation. Okay, so the next is the novel pathogenic mutation. Now novel means it's new. So this kind of sequence variation is actually not previously reported, but is actually expected to cause the disorder. Now generally the truncation mutation or the novel nonsense or the frame shift mutation, the missense mutation which affects this translation initiation codon, or the splicing mutation that altered the invariant AGGT boundary, they all belong to this particular category of novel pathogenic mutation. Okay. The third is actually a novel variant that may or may not be disease causative. So this means that this sequence variation is not previously reported, but it has uncertain pathogenicity in genes that is known to cause this inherited Mendelian disease. And this type of variation include these following mutations, that is the missense variant or small inframe deletions or duplications, and for which the generally effect is on protein. Okay, and apart from that you have the exonic or intronic variants and the putative splice variant or the variants in regulatory sequences. Now the fourth kind of variant is actually a novel variant that is likely to be benign. So this means that this sequence variation is not previously reported, and it is probably a benign change. Now that means synonymous changes and deep intronic changes beyond 20 base pair of exon intron boundaries are generally found in they fall in this category. Okay. Now so this is a novel benign. So apart from that I should have another, that is the non-benign variant. So that means this sequence variation is previously reported, and it is a recognized neutral variant. That means, for example, a variant that is reported in the database of SNPs with a frequency that highly suggests that it's a benign. That means that generally to be benign, the average heterozygosity must be less than 2%. Okay. and the total chromosome count must be greater than 100 and all. And the last one is actually a reported variant with unclear pathogenicity. That means this variant is actually reported as a mutation, and this is done by based on observation in a single patient or in a single family, or in a by a study that is found in a population association studies, and they actually have no functional studies that can address this pathogenicity, and it is not reported in the DBSMP as a rare variant with average heterozygosity below 2%. Now please note that this category does not completely correlate to category 6 of the ACMG guideline. In the ACMG guideline, these are actually the variant that is found in association studies. And in this particular classification criteria, the ACMG variant category is actually 6 variant is actually classified into either category 5 or category 6, and that is based and this thing actually the known benign and the reported variety variant with unclear pathogenicity, it is actually based upon the allele frequency that it can be found in each individual variant, right? Now this just wait a second. Okay, now why do we now we need to interpret this sequence variations, right? Now proper classification of this whole sequential variants into these different categories, they facilitate the interpretation of the clinical significance of the variant. And generally this is the algorithm for that is used for sequencing variant classification. Okay. Now generally the alleles that is found in classification 1 and 2 is categories 1 and 2, they are considered to be a bona fide deleterious, and they are actually disease causative. Because the mutations in these two categories must be included in the patient's report with a discussion as to the pathogenicity and the proper reference to publication in which they are reported. Now generally when you find this kind of mutations in the patient, then parental studies might be recommended in order to confirm the findings, because since if these are actually reported pathogenic mutation or it's a novel pathogenic mutation, then they might be genetically they might be genetically transferable. So if you can find that same kind of mutation in your parents, and if you find that your parents also suffer from the same kind of disease, then you are confirmed that this particular variant causes this disease, and it might get transferred to your child also. So it must be before, you have to tell the parents beforehand that whenever you are going to get a child, he or she might succumb to this particular disease, right? And for that the parental studies are needed, because sometimes if it is this kind of mutations are found in the autosomes, then and if it is found in the woman, then that might skip a generation, right? So that's also a probability. So you have to look for everything to and discuss with the patient like this. For example, if you have a homozygous mutation, okay, you have idea about homozygous and heterozygous. So if it's a homozygous mutation, then parental testing is necessary in order to confirm that each mutation is heterozygous for the finding, as opposed to being in one allele, and that has the failure to detect the other parental allele for technical or genetical reasons. And if two heterozygous mutations are detected, then the parental studies facilitate the determination of the phase of these two mutation. Phase means cis or trans mutation. and the this generally the familial targeted analysis should also be provided for the at-risk family members. Now, variant in this category, this fifth category, the known benign variant, these are actually benign polymorphisms that are common in a healthy population.
Okay.
Now, this benign variant may or may not be included in a patient's report, but they should be available upon request. But these equation variants that are found in this third, fourth category, these are considered to be the variant of some unknown clinical significance, right? Because they have some unclear pathogenicity, and this category 6 also falls under that particular thing. And the assessment of the clinical relevance of this unclassified variant is not straightforward, and it can be extremely challenging. Now, there are actually several resources such as variant database and in silico prediction algorithms that can be used to provide evidence for or against the pathogenicity of these unclassified variants. And these are actually done to aid in the genetic counseling. Now, the report of this unclassified variant, they also should be issued to appropriately trained healthcare professional, because it is essential to discuss the result with the clinical geneticist and to request additional specimen from the patients and family members for additional testing in order to facilitate the interpretation of this unclassified variants. But you know that sometimes the testing of an unclassified variant for predictive context, for example, to find a prenatal diagnosis, is generally not recommended, because in prenatal diagnosis, since you don't know what that mutation gives rise to, even if you find that kind of mutation in a fetus, you cannot confirm anything. But that might panic the parents, and panicking is really bad, because when you panic, you tend to take some irrelevant decisions, right? So this is one of the things that, therefore, whenever you have something that is not very unclear or you are not sure of, you should not tell that to the patients. And apart from that, it is also used for the deletion and insertion interpretation, because the size of the deletions and insertions that is detected by the sequence analysis is relatively small. And it is when you compare it to those array-based detection or the fish assay, fish, the fluorescence in situ hybridization.
No.
Okay, in fluorescence in situ hybridization, what we do, so tomorrow I'm going to show how fish works, okay? Just because if I tell you, you're not going to understand. I'm going to show you a picture and then make you understand, okay? And if this observed deletion or insertion, if those are not previously reported, but you get the results in the alterations of the protein reading frame of a gene that leads to a frame shift or nonsense stop codon, then it must be classified as a deleterious mutation. But if you have, suppose you have a deletion and insertion that is novel, that has not been reported, but you find that this particular deletion or insertion results in an in-frame insertion or deletion, then depending upon the size and location, it may or may not affect the function of a protein, because a small in-frame deletion or insertion is generally classified as an unclassified variant. And therefore it is really important to check if that particular indel involves an important functional domain or not, because sometimes if that particular deletion corresponds to an important functional domain, suppose it corresponds to an active site of a protein, then it will have some impact on protein function, right? But if that particular domain does not fall into the active site, but it actually falls into a very in a core hydrophobic domain that is generally present inside the structure, then it is not going to affect the protein function. It might affect the protein stability and everything, but it is not going to affect the protein function, right? Understood? Okay. So this is the overall algorithm or thing that we should do for sequence variant identification. So what do we do? First of all, we have a sequence variants, right? So the first thing we can do is first check the database of single nucleotide polymorphisms. Now in that, if that particular kind of thing is already mentioned, it is if it is already reported, then it will be present in the databases.
Yeah.
So there will be two results. One, either it is present in the database or it is not present in the database.
Yeah.
So if it is present in the database, then there are two kind of things. That one is, yes, it is reported in the databases, but it has the heterozygosity is more than 0.04%, and sometimes it might not be present in the databases, but as it has a heterozygosity, or it might be present, but the heterozygosity is too low. It is too low. And if this green thing happened, yes, it is reported, then it is likely to be a benign variant, because the heterozygosity is only above 0.02%, right? But if it happens that no, this particular kind of thing is not present in the single nucleotide polymorphism database, then we have to find other databases if those mutation is present or reported in other databases or not. And even if that particular kind is not reported in anywhere, then it's a novel variant, right? And if it becomes a novel variant, then it will be an unclassified variant. That it is not, the phenotype is not linked. The particular phenotype is not linked with this particular genotypic changes. And suppose you can also go for the nonsense and find out the nonsense frame shift or invariant splice site mutations. And this particular kind of thing, if there are any kind of this nonsense or frame shift mutation, now generally these are pathogenic mutations, right? These are pathogenic mutations. So even if you find a single kind of thing, you are going to report it as, yes, it's a pathogenic mutation that you have, and it might cause some disease into you. That's a high level of scrutiny. And apart from that, you have to find out by doing this and that, and you have to classify, and you have to resequence all of them to find out if you have missed anything or not, and then base your idea depending upon the because you have to then check for your parents, check the DNA from their parents, or check the DNA from their offsprings, just to make sure that those kind of thing tallies with the phenotype and genotype. Okay. And apart from that, you can also use this for evolutionary conservations also, because it is seen that because the analysis of this conservation of this amino acid at a specific position of a protein is actually a first step to infer the structural or functional importance of this amino acid residue. Because if you see that a particular amino acid is remaining unchanged throughout the whole evolution, then you know that particular kind of mutation is really pathogenic. You must have that amino acid at that exact location. If you change that amino acid with that exact location, the gene is going to, or that particular person or that particular organism is going to die. So that's why this is really important to find out the specific position of the amino acid in that particular protein. And the protein sequence that contains the amino acid of interest, they should be used as a query for analysis using the BLAST website or some other website that we're going to study in proteomics section. Okay. And if you have some orthologous proteins from various species throughout the evolution, then it needs to be selected and retrieved. And there are different biological species that should be selected from the phylogenetic trees that represent the similarities and differences in their physical and genetical characteristics. But as a minimal standard, the alignment should include the full length sequence of generally all the genes, in which at least 5 are from mammalian species. Now generally, if you have Homo sapiens, then you can use Homo sapiens, then the gene from cow, the gene from dog, gene from mouse or rat, gene from chicken, frog, fish, etc., etc. And apart from that, you also have another server, the Cluster W is a multiple sequence alignment tool, and is actually recommended for multiple sequence alignment. Okay.
PCR based.
Okay. Now you can see that generally, as we have seen starting from the 2D section, we've seen that in the 2D section what we used the emulsion PCR or bridge amplification method, right?
Yeah. Mm-hmm.
Both are a type of PCR. We generally use PCR for amplification of our DNA.
Yeah.
But it is now seen that this PCR actually it produces and it introduces some series of errors into the into your system. Why? Because first of all, when you are using PCR, you have to use the Kleno fragment, right?
Yeah.
And in the Kleno fragment, you lack the proofreading activity of your polymerase. Now, as the proofreading activity of your polymerase is lacking, it is bound to introduce and make some errors.
Mm-hmm.
And as it introduces, and the error rate is really high for the Kleno fragment. It is really high. So this PCR based errors and this PCR made errors are actually, they generally crops up into the system, and these are not human made. This is not made by you. So it is not up to you to rectify this mistake, and you can never rectify this kind of mistake that happens as a by the PCR amplification procedure.
Mm-hmm. Yeah.
So generally you see that when we moved into the third gen sequencing or even in the solid sequencing, people tries to avoid these PCR based methods, right? And therefore we only try to maintain the either by the ligation procedure or by using the general polymerase, the whole polymerase. We use the whole polymerase to and make up the maintain the same chemical environment in the ZMWs on into the in the microwriter plate, and do perform the DNA amplification or replication to find out the sequencing platform. Because what happens is that I'm going to make you understand you're going to learn how to develop primers in the genomics section, how to develop primers and all. In that particular thing, you are going to understand in much detail, but how to do that, what to do, and why to do. But generally learn that you see, when we develop some primers, the primer lengths are minimal. It never exceeds 10 or 12 nucleotides, the type primer length. It just wait a minute.
Yeah.
So whenever you use a very short primer of 10 to 12 base pair, what happens? It fails to identify the exact location, right?
Yeah.
And it might bind to a different location of interest. And if it binds to a different location of interest, what will happen? It will amplify a different region. So generally the idea is that you must develop or generate a primer more than which the length should be more than 17 base pair. Okay. That's the basic idea. Now this also has several problems, such as that when you have a primer that is more than 17 base pair and you are only allowing it to bind to a complementary site, sometimes it fails to bind. Why? Because the length is really high. So whenever suppose a 5' end bonded to that particular complementary region, now until the time when the 3' end also binds to that exact location, the 5' end gets destabilized and it opens up. So this kind of primer binding errors also crops up, and these are something that when you are carrying a minor PCR of a short sequence, it's good. But when you are sequencing for the whole human genome, then even a single error can enhance to a very million nucleotides, right? Because I'm amplifying it almost 10 to 100 to 2000 times. So that's how this conventional PCR based methods are generally now people are avoiding these PCR based methods, and they're going for either the whole,
ligation.
yeah, either they're going for the ligation based method or they are using
Poly.
original polymerase to replicate the whole DNA. They're not amplifying the DNA, they are using the whole DNA and they're using a
Replicate.
particular set of polymerase and then they are asking the polymerase to replicate the whole DNA. Bas, that's all. I don't
Okay.
need multiple sequence because multiple sequence. Why do we need to amplify the DNA? Just to so that we know that when we if we have several fragments of DNA and if we read all the fragments and if I align all the fragments one after another, then even if there are some fragments in if there are some errors in 1, 2, 3 or 4 or 5 fragments, the rest of the 10 fragments have no errors at all. So we can nullify the errors by the quantity of fragments, right? So that's the
Yeah.
basic idea. But that does not work because people have already found some ways to go behind that particular thing. Okay. Now, okay, just a minute. Okay, so now I'm going to go with some this single DNA molecule barcoding method, and it has the application for molecular haplotyping and DNA mapping by this NGS. Now, this
Yeah.
success of this human genome project is largely due to the continuous development of this Sanger sequencing method, right? And that has been
Yeah.
done through parallelization, automation, miniaturization, by developing better chemistry or better informatics. Now, as the workhorse of this human genome project, this Sanger sequencing method, it has actually dominated this DNA sequencing field for nearly 3 decades. However, its serial electrophoretic processing of this DNA sample through the microchannels will not be able to compete with the newly emerging massively parallel sequencing technology like ABI Solid, Flexa, this genome analyzer, and all. And this newly emerging technology can be roughly grouped into 2 categories based on the detection method. Either it is sequencing either by the ensemble detection or by single molecule detection, right? That it does by smart and all.
Yeah.
Now, since these multiple DNA copies are needed in ensemble detection, the genetic information such as this haplotype and RNA splicing pattern is lost during the process. Okay. Now, here what you're going to do, I'm going to make you understand that how we can employ this NGS to find out the DNA mapping, and
Okay.
this is known as a barcoding method. Okay, so now yes, so this single molecule, so I am talking now this smart technology. They use this single molecule detection, and this single molecule detection, it actually holds great promise to recover this haplotype information.
Yeah.
But the read length of this current single molecule sequencing method is very poor. It's merely 50 base pair or less, and which is far shorter than the average distance of 1 kb between two SNPs. Right? You know that, right? Generally, between two SNPs, there is that average gap is between 1 kb because
1 kg.
two SNP does not happen to uh, two neighboring atoms or two neighboring nucleotides does not get the same SNPs. Generally, the SNPs are separated by a 1 kb of distance, generally.
Okay.
And just like its predecessor, that Sanger sequencing method, the critical genetic information such as this haplotype and RNA splicing pattern is difficult to obtain with this NGS system. Now, the future sequencing technology, they should resemble the DNA replication system in every cell, and simply taking apart all the chromosome and reading each base of the chromosome from end to end. And that's what the smart technology tries to do. Okay, right?
Yeah.
Now, in this initial attempt towards this final goal of reading each base of the chromosome from end to end, people have developed a single DNA molecule barcoding method that can obtain the genetic information across the genomic regions of over 10 kb.
Okay.
Now, this barcoding strategy is based on direct fluorescence imaging and localization of multiple sequence motifs. or even you can do it for very polymorphic sites on a DNA molecule. Now, individual DNA, genomic DNA molecules or long-range PCR fragments, they are labeled with a fluorescent dyes at very specific sequence motifs or polymorphic sites. Okay. So suppose what's happening in this particular case, you can see I have this particular PCR primer or PCR fragment, and I have labeled it with a fluorescent dye. Okay.
Yeah.
So now this labeled DNA molecules are then stretched into a linear form on a modified glass surface and imaged. They are imaged by using the TIRF microscopy. TIRF microscopy means total internal reflection fluorescence microscopy. And by determining the position and color of the fluorescent levels with respect to the DNA backbone, the distribution of the sequence motifs or polymorphic sites can be established with good accuracy. And this is in a manner similar to reading a barcode. And people have successfully applied this single DNA molecule barcoding method in DNA mapping and molecular haplotyping. Okay. So now in this DNA barcoding method, one has to determine the positions and the colors of the fluorescent level that is found on the DNA molecule being analyzed. Now there are generally 5 critical steps involved in this particular DNA molecule analysis. Okay. And what happens? So generally, what happens is that in this particular thing, the general idea is that you have this particular primer. You have this particular primer. Okay, and you fix these primers on a flow cell. Okay, or onto a matrix. And once I have fixed these primers onto a matrix, and if I add my chromosome or the whole DNA to it, so what will happen? They will obviously bind to a exact location. They will bind to the complementary sequence. And once they bind to a complementary sequence, I can see the fluorescence. And by recording the fluorescence, I can also map. And by taking since I am using a microscopy, I can map the exact location of this sequence into the on the chromosome. And by doing this, I can map exactly how your sequence looks like. And so we know that these barcodes are really different for each unique samples, right? Each barcode is different. And similarly, if we do this, since we have different number of SNPs, a different restriction endonuclease site and different polymorphic sites, this sequence will also be different in our body. Right? And so it will vary from person to person. And that is the basis of this molecular barcoding. So to find out the difference between each different person. And so we can simply isolate and take people and name people that yes, it is different from him, he is different from him. And like this, if we can map the genome of the entire population, the idea is to actually map the entire population, then what will happen? Even if someone even does a crime and all, if we have these records, if you already have this kind of things in your record, then it will be easier for the forensic team to catch the real criminal. But overall, you can also use this to find the genetic defects, because some genetic defects are, suppose if it's a breast cancer, then you are going to have some mutation in the BRCA1, BRCA2 gene. And the kind of mutation that is found in BRCA1 and BRCA2 are different, because the first kind of mutation generally leads to a benign cancer, and that's one cancer that people that's not serious, right? Once you have a metastasis cancer, a second kind of mutation leads to a metastatic cancer, and that's highly risky. That's a high risk group, right? So if you can barcode and find out this kind of pathogenic mutations, and if you can simply trace back those kind of mutation into the person also, and it is much easier, because we are not sequencing the whole genome. What I am doing, I am simply mapping that kind of location, a kind of mutation into my chromosome. So just taking your chromosome and doing a microscopy will simply efficiently tell you whether you have that kind of mutation or not.
Yeah.
So this is much easier than sequencing also, but it is really important, and you can find out about the different kind of mutation in you, and you can already take some precautions and all. Okay. uh, okay, so we are stopping here, and tomorrow we are going to discuss the basic steps that are, and that are actually the critical steps that are required for this data molecule barcoding methods and all.
Okay, sure.
Okay. Okay. And I gave you a call, that's my number, so you can simply
Okay.
call me anytime you want. And tomorrow morning by 8 o'clock I will let you know that when I'm going to take the class.
Okay, sure.
Okay.
Okay,
Okay, fine.
got it.
Okay, and
Thank you.
I will be sending you the schedule of this week's class soon. Okay,
Okay.
I'll mail you.
Sure.
Okay, sure. Thank
Thank
you.
you. Good
Yeah,
night.
surely. Thank you. Good night. Hmm.